In this video, I'm going to talk about thermotolerance, specifically thinking about tolerance to high temperatures and to cold temperatures. And I'm going to focus largely on characteristics that are important for trees that grow in seasonally cold climates. So we're going to start with cold tolerance. I'm going to cover three different topics in this area. And the first topic has to do with plant hardiness and the ability to tolerate cold and freezing temperatures. So many of you may have seen this plant hardiness zone map. It's a little pixelated in this version, but hopefully you can see for Minnesota that we tend to be in the low temperature zones compared to places to the south of us. If you look specifically at Minnesota, it's a little bit clearer. You can see that northern Minnesota, we're in part of zone 3 that reaches temperatures of negative 40 Celsius, whereas in the southern part of the state, we have little patches that are in zone 5A that only don't even reach minus 30 Celsius. And minus 30 Celsius, for those of you who tend to think in Fahrenheit, is about minus 20 Fahrenheit. So how do plants survive going down to negative, below freezing temperatures? Well, there's a number of aspects related to reaction to cold temperatures, and it depends on the level of cold, to what degree any individual is using any of these mechanisms. I'm going to walk briefly through this table. Basically starts with cool temperatures in the fall. Some parts of the U.S. never get colder than this, so this preparation stage is about all plants are able to do. As temperatures get towards the freezing point of water, trees tend to alter their cell membranes by changing the composition, putting more unsaturated fats, which means as the membranes get colder and colder, they don't become stiff. You can think a little bit about different oils. Liquid canola oil, if you put it in the fridge, stays liquid. If you have ever put olive oil in the fridge, you'll see that olive oil solidifies. And basically, that olive oil has more saturated fat, and so it is less flexible as it gets cold. So cell membranes become unsaturated in order to stay flexible. That's these cell membrane changes. And then a number of proteins and other factors are put into the cell walls that also influence flexibility and also influence whether or not ice crystals form, because basically if you're going to freeze, freezing needs a ice nucleation site. So some particle around which to start to form ice. So these are early changes, and for species that don't go below -1, those species, this is about all they do. Down to about -5°C, plants employ what we might call an anti-freeze strategy. So basically they dissolve different solutes into the water that's inside the cells, and those solutes prevent ice crystal formation by lowering the freezing point of that liquid inside the cells. But this doesn't apply to much lower than -5. Once you get below -5, you get into a situation where the cells in the plant really need to dehydrate. They need to move that water that is inside the cell outside the cell and dehydrate. So they need to take that intracellular or symplastic water and move it extracellularly or into the apoplast. What tends to happen with this is that these crystals start forming outside of the cell. The water that gets moved outside of the cell occurs outside of the cell walls, and this does not break the cell. Below -15, any water that's left in there and it is in a super cooled state. And so it's held in small areas around dissolved particles in the water. So there's still these solutes in the water that's allowing this kind of anti-freeze super cooling to happen. This is a very unstable state, and so a small change in or the introduction of some impurity into the water that's left will lead to rapid freezing. So this is a period where whatever is left in there is in this supercooled, fairly unstable state. And then once you get below minus 30, basically the tissues are freeze-dried. So the cells are completely dehydrated except for water that's part of molecules, and all the water outside of those cells in the apoplast becomes crystallized and frozen. So basically the cell itself is freeze-dried and the water outside it is frozen. So I have some images to show kind of the difference between a freeze-dried and a cell where you've had ice crystal formation inside. That'll show in a little bit. So cold hardiness, there's actually 3 different groups. There's groups with limited hardiness that basically can use these strategies that get them down to minus 15. There's species that can deep supercool. So these are these that can go to minus 40 into this supercooled state. Oaks are a species that can do this. And then there's others that can tolerate this freeze dehydration. So there's this set of species that basically can get below minus 40. And minus 40 is really this, it's kind of a magic point. It's a point where Celsius and Fahrenheit converge. 40 C equals minus 40 Fahrenheit. But this is also this point where supercooling no longer works, and you move into this, everything is frozen and dehydrated, and only certain species can withstand that kind of stress. So here's some images. This is the ice particles in the symplast on the left-hand side over here. These are these ice crystals. They're inside the cell, and basically the fact that these ice crystals have formed has basically destroyed the entire contents of the cells, and the cell will be dead. Here's an example where all that water moved outside the cell. You can see the cell is much smaller than its original footprint. It's dehydrated, and these ice particles are now outside of the cell. And this cell is likely to be able to rehydrate in the spring when this water melts and is taken back up by the cell. So this process doesn't happen just automatically. There is a process of acclimation and going into dormancy that occurs over the course of the fall in north temperate zones. So what this graph shows is basically the date on the x-axis, and then the lowest temperature that a species can survive on the y-axis. And you can see in early September they can survive slightly negative, but not very negative. So this is in that cell membranes have shifted configuration, but not in a acclimated stage in which they are employing these other mechanisms. Generally, a first frost or a first kind of set of cold temperatures triggers a bunch of gene activity that moves the plant into even a deeper acclimation. This is the state where they're able to basically develop these anti-freeze characteristics, and so the water gets cooler and cooler, is basically super cooled, and is able to withstand lower temperatures. Then you have a longer period that they stay at this, and then you get their first hard frost. So up here, this is cold, chilling temperatures, probably like we had in the last week or so. Then we have our first hard frost, perhaps in mid-October, and this starts basically a cascade that moves the species into a state where they're able to enter this extremely deep dormancy. And this is a trace actually from dogwood, and dogwood is one of the this third group of species are able to go well below minus 40, completely dehydrate the cells that are there, complete extracellular water freezing, and able to withstand extremely low temperatures. So the second aspect of this, so our first aspect is really this cold hardiness and the ability to employ these different mechanisms with which to withstand freezing. The second mechanism has to do with xylem. Once again, we're back to the xylem issue like we had in, like we talked about with drought tolerance. And again, just a reminder, xylem are these dead cells through which water flows. And it's very important that these tubes do not get filled with gas, because that breaks the water flow and in a process called embolism or cavitation. And one thing that happens with freezing in xylem cells, you have these water-filled xylem cells in the winter and they freeze. And if anyone's ever looked at a frozen ice cube or the frozen lake, there's air trapped in there, right? So when the water freezes, gas is emitted and you get air bubbles that are trapped in those in that ice. When that ice thaws, there's the possibility for those air bubbles to coalesce into a large enough bubble that would fill one of these vessels with air and cause embolism or cavitation. Now, the extent to which this coalescence of the tiny air bubbles happens is dependent on how many freeze-thaw cycles a tree goes through in the fall and then in the spring as it starts to warm up. And it's also a function of how big the xylem vessels are. So this large xylem vessel is much more likely to be embolized by this freeze-thaw gas bubble formation than this very small trachid cell here. And if you want to know more about why, I'm happy to talk to people outside of class about that. And so this is a cross-section through a gymnosperm. It's all trachids. Gymnosperms tend to have low susceptibility and low levels of freeze-thaw embolisms. This is part of why these conifer species that are trachids only tend to dominate as we move into colder and colder and colder climates. In contrast, something like an oak, which is ring porous, this is a ring, they have these big xylem vessels. By springtime, for the most part, ring porous species like oak are completely cavitated. They cannot move water. And so before they can actually move water to new leaves, they must grow new xylem, a new growth ring in the spring to move things. So this is part of the the differences between species in terms of their ecology related to cold and tolerating cold. Gymnosperms can become much more active, not only because they have evergreen leaves, but because they have functional xylem in the spring, compared to something like an oak, which tends to be one of our later leafing species because the xylem needs to be basically rebuilt, regrown, so that they can functionally transmit water. Okay, the third aspect has to do, again, with dormancy. So this is a picture from a paper written in 1920. This is the influence of cold in stimulating the growth of plants by Frederick Coville. I love that he was the botanist in charge. I would like to be the ecologist in charge of something. That might be fun. So basically he did an experiment where he, this is blueberry, he grew a deciduous blueberry, and one branch he stuck outside of the greenhouse for the whole winter, and the other branch he left inside the greenhouse. So take a minute to think about which of these two branches, this is the next spring, which of these two branches do you think was outside of the greenhouse for the winter? If you chose the right side, the leafed out side, you're correct. What this experiment basically showed is that buds in temperate species require cold to leaf out. So this branch over here didn't receive any winter chilling, no winter cold, and it failed to leaf out, compared to this branch which received winter cold and leafed out in the spring. So this is a very common aspect of dormancy in temperate trees, and is one of the kind of mechanisms that influence the tolerance of species as well as where they occur on the landscape. If you grow red maple from Minnesota, for example, in Florida, and it goes into dormancy in the fall, it's never going to leaf out again because it doesn't have that cold signal. So this is kind of part of that adaptive suite of characteristics related to cold tolerance. This is also true, and you may have heard about this more for seeds, that many seeds have a winter chilling requirement or stratification. And so one of the problems with plants setting seed throughout the summer and into the fall is how do they know when to start growing again? If they germinate and start growing in the fall, they're going to be subjected to very cold temperatures very early on in their life cycle. And so many temperate species employ winter chilling as a cue. They must have a certain amount of chill or they won't germinate at all. All right, so let's move on to heat tolerance. Heat tolerance is a little bit of a different beast. This is an image of a project I'm involved in where we're using infrared heat lamps. This is an infrared camera. These are really hot. They're warming trees, and we're basically looking at how do trees deal with warmer temperatures. So heat tolerance is partly related to physiological responses and the kind of relationship of physiology to temperature. And so if we look at the x-axis here, we have temperature, and then we have gross photosynthesis. So this is all the carbon taken in by the plant, and it rises to some optimum. So there's a set of optimum temperatures for gross photosynthesis, and then as we get hotter and hotter, it drops off largely because of basically tissue denaturing, proteins denaturing at high temperatures. Respiration has a slightly different response, increasing exponentially more or less with temperature, and then at very, very high temperatures, respiration or carbon loss falls off a cliff, basically. Net photosynthesis, the carbon that the plant keeps and is for use in other aspects of their physiology and biology, the carbon that they can use for growth is what's the difference between these two, this gray area in between. And you can see that at low temperatures you have low photosynthesis, you also have low respiration, but the difference is quite small. So low temperatures, carbon gained and able to use for other function is quite small. In this case, this is kind of a best case scenario, or maybe a scenario for a place that has kind of temperatures between 20 and 30. Optimum occurs here where basically high levels of photosynthesis and respiration still remains rather low. As we move out here, even though photosynthesis remains rather high, respiration continues to rise. So the level of basically carbon that's used for growth and other functions declines to some point where respiration exceeds photosynthesis being brought in. So what an individual species temperature response profile looks like is very important in terms of the kind of temperatures at which it thrives and tolerates. So here's just a figure showing photosynthesis. This is really percent of the maximum observed over a range of temperatures. So you see this kind of optimality curve where it rises as temperature rises and then it drops off. And this is a cartoon, of course, so things don't all look like this. But you see that if we think about this as species 1, 2, 3, and 4, that species differ in their temperature optimum, with species from warmer climates presumably having higher temperature optima than species from colder climates. So we could think of these two as temperate species, these two as tropical species. And you can imagine if your prevailing climate in a site is, say, 12°C, something like that, that different species are going to basically have higher rates of photosynthesis and potentially be better competitors along different parts of this gradient. So here or at lower temperatures, species 1 might have an advantage in terms of growth and competitive ability. As you get a little warmer, species 2 gains advantage. As you get a little warmer, 3 gains advantage, and at the highest temperatures, 4 has the advantage in terms of photosynthesis, and to the extent that that translates to growth and competitive ability. Now, this isn't a fixed property. This is a graph from a Californian desert shrub. And basically, this is the photosynthetic rate, this across a range of temperatures for an individual shown in blue here that was grown at low temperatures. And then that individual was moved to a higher temperature, grown for a little while, and you can see that this optimum shifts. So species have some flexibility here, and potentially the degree of flexibility that a species have influences the range of temperatures to which they are well adapted. Respiration can also acclimate. So here's a kind of a cartoon looking at respiration of a leaf measured over a range of temperatures that had been grown in, say, an ambient climate conditions in northern Minnesota, and it has a particular shape. Then we take a leaf on a plant that was growing in slightly warmer conditions, so plus 3.5 degrees Celsius. Do that same curve, and what you can see is that it's shifted downward. Oops. It's shifted downward, and this downward shift basically represents a downward acclimation of respiration, such that at any given temperature that species loses less carbon. So this is kind of a way to adapt and acclimate your physiology to warmer temperatures. So for trees, actually, the response to heating trees isn't really about the capacity to tolerate those ultra-high temperatures. Ultra-high temperatures, they're rare, and especially those that cause physiological dysfunction. So physiological dysfunctionally high temperatures are in like 45 to 60, which you basically only get in the hottest of hot deserts, and trees don't occur there. So for the most part, trees aren't dealing with super high temperatures. What they are dealing with are this range of temperatures, and so response to heat in trees and how they interact with others really has to do with the ability to compete with each other, and those that may be better adapted to warmer climates win out in this kind of competition. The other thing that I didn't talk about here, but harks back to Monday's videos, are that actually when you get to these higher temperatures, what tends to start to dominate in terms of heat response is actually the associated drought responses. So high temperatures tend to lead to high evaporative demand and potential for water loss from transpiration, but also from evaporation. They tend to be associated with low rainfall periods of time. And so the capacity to tolerate drought actually ends up being one of the major issues associated with species tolerating heat. So if you go back to the drought tolerance lecture, you can review those drought tolerance traits and think about those as basically also characteristics associated with high temperatures, since drought tends to follow high temperatures. So in summary, cold tolerance in trees is related to their cold hardiness and their ability to withstand increasingly cold temperatures, their xylem function and how sensitive they are to freeze-thaw cycles that occur in spring and fall, and as well as dormancy release requirements. Heat tolerance, largely in temperate trees, has to do with their physiological optima and the flexibility of photosynthesis and respiration, largely relative to their competitors. So those that are able to shift their physiology to better match prevailing temperatures presumably will gain more carbon, grow more, and have a competitive advantage And then the other key aspect of heat tolerance in temperate trees is actually their ability to withstand drought, since heat and drought tend to go hand in hand.